Our second American Heart Association Allen Distinguished Investigator, Dr. Jeffrey Holmes from the University of Virginia, will be talking about information storage and retrieval in the cardiac extracellular matrix.
Thanks, Kathy. So while we're waiting on slides, I'd like to add my thank you to the Allen Frontiers group and to the American Heart Association. It's been really fun with Sunil to be the part of the first wave of the AHA Allen collaboration, and then I'm really excited because right after lunch we'll get to hear about several of the new projects that are being funded also as a collaboration between American Heart Association and the Allen Frontiers group. All right, so my lab studies heart mechanics and disease, and so when I think about myocytes, the cells that do the main work of contraction in the heart, I think of them like this, which is the way they look in vivo: long, skinny, aligned with one another, and if you zoom in a little bit, having lots of little striations called sarcomeres that are the actual contractile units. But if you take the myocytes out and you put them in culture to study them, you usually get something that looks kind of like this: spread out, not a very good contractile apparatus. Very, very different from what we have in vivo. And people have thought about ways to force those cells to do something in culture that's a lot more in vivo-like. In this case, by micro-patterning to try and force the cells to take on a more in vivo shape, or in this case, by surrounding the cells with a cocktail of different extracellular matrix molecules and then mechanically and electrically stimulating them over time to get them to evolve into something that looks in vivo-like. This is all work by people who are doing cardiac tissue engineering, and it's turned out to be really, really hard to make cells that are even a little bit in vivo-like. What's interesting is if you contrast that with another group that cardiac tissue engineers have been taking. This is all from Doris Taylor's group, where they took a normal rat heart. Here again are the myocytes. This time they're not fluorescently stained. They're just stained with H&E, but you can see that they're highly aligned, and they're all the pink things in here. They decellularize, so wash the cells out with detergents that leave behind the extracellular matrix, and you see now the heart's completely clear. Then they take cultured cells and infuse them back into this scaffold. So first, the cells, endothelial cells that would normally line the blood vessels in the heart, and sure enough, they repopulate the insides of the blood vessels in the decellularized heart. And then you take myocyte-like cells in culture, and you perfuse those in, and sure enough, they reoccupy the spaces where the myocytes once were, and they elongate, and they form sarcomeres again. And so what I think is really interesting about comparing these two tissue engineering stories is that it reveals something that the more we look, the more true we find it to be about cells in the heart, which is that so much of what makes those cells the cells that they are is the instructions that they're receiving from the extracellular matrix around them. And not just structural in determining the shapes, but also biologic signals in two forms, in the sense that by attaching to the matrix, you're activating intracellular signaling pathways that affect the biology, but then also in the sense that Sunil talked about a little bit, that we're finding that all these little fragments of extracellular matrix molecules that get cleaved off by various proteases also have signaling functions, and themselves will then immediately instruct the cells to do different things depending on the amount of degradation that's going on. So what makes all this even more a little appealing to me as somebody who studies heart disease is that we know that the half-life of the proteins in all of the cells is on the order of days. So those sarcomeres that I showed you before, everything in a sarcomere turns over every couple of weeks. But the half-life of a lot of the extracellular matrix proteins can be on the order of months, or in some cases even years. And furthermore, we know that over those long time periods, especially in conditions like diabetes and inflammatory states that predispose you to heart disease, that those matrix molecules accumulate post-translational modifications over these very long time courses. And so the overall premise of our project is that in order to understand heart disease that's evolving over very long time periods, we need to understand more about cell extracellular matrix interaction, and in particular about how one cell may modify the matrix in a way that then affects the behavior of the next cell. So to do that, one of the techniques that we're harnessing is something called STOMP, which is short for Spatially Targeted Optical Micro-Proteomics. And the basic idea of STOMP is that you come in and you label something that you're interested in with a fluorescently tagged antibody. In this case, we're showing two examples. The orange is if I were interested just in collagen fibers that are in the heart and wanted to label those and focus on those. And then the red is if I was interested in a particular cell type and wanted to go in and label that particular cell with an antibody against a cell marker. Once I've labeled those, then we're going to come in and we're going to map all of the locations of those fluorophores in a confocal, and then we're going to photo cross-link 1 cubic micron around each one of those fluorophores with a tag. Then we digest everything and we pull down everything we tagged. So now I've just selected for things that were in the regions that I labeled with my fluorescent antibody. This just showing an example of that with, in this case, we're tagging for alpha smooth muscle actin, which I'll talk a little bit more about later. And we've got a couple of cells in here that are expressing that. This is a zoomed out view where this is the central region is the zoomed in part here, which will be important in a second. Here we then make a mask showing the things that we've thresholded out. We cross-link. In this case, we're cross-linking biotin. And so now the green is showing you where the biotin got cross-linked. So those are those 1 micron cubic voxels. And this is just to show the zoomed out that we're not accidentally cross-linking any biotin any place that we didn't want it, and then an overlay to show that it ended up in the right place. Because we're subselecting little bits of tissue on frozen tissue sections, we're going to have to do a lot of this in order to get enough material to analyze. And so this is automated. We can go in and on a tissue slide, select the region of interest. That gets automatically tiled. And then this process is repeated automatically for each of the locations in the tile. First, imaging where the label is, then making the mask, then doing the photo cross-linking and scanning through the entire slide in that way. All right, so here's an example. As a test case for developing some of the methods, the disease we've been working on the most is myocardial infarction. And the reason we picked that is because we know that the extracellular matrix is changing very rapidly. What happens after a myocardial infarction is muscle in a region that's affected dies. It gets replaced by scar tissue. And so you have a big inflammatory reaction and then a big fibrotic reaction. So we know that the things that we wanted to look for are changing dramatically. Here, this is a zoomed out view from, it's a piece of tissue that's several millimeters on a side from rat infarction tissue at 1 week. This is the alpha smooth muscle actin label in the post-infarction scar. What that mostly is labeling is fibroblasts, but you also get smooth muscle cells in big vessels. So those are those sort of oval-shaped things you see here. And then this CD68 is a marker for macrophages. And so the green here is macrophages. And you can see if you compare these two that there's a lot of macrophages and fibroblasts at this time point after the myocardial infarction, but that the distribution of those two cell types is quite different in this case. All right, so then little bit of methods. So we pull everything down, we digest, we send for mass spectrometry. That part is pretty traditional, and the peptide identification is also pretty traditional for those of you who are used to doing mass spec. One interesting thing that comes up is how to normalize, and that's because we're intentionally trying to compare the matrix right around different cell types that we expect to have very different surrounding matrix compositions, and then we're also comparing over time at times where we expect the matrix composition to be completely different. And so what I'll show you today was actually normalized using what you might consider an artifact, which is the peptides from the streptavidin beads that we use to pull everything down. Because we always use the same quantity of the beads, the same for a similar amount of tissue that we are analyzing, that actually turns out to be one of the things that stays consistent across all the time points and across all of the different cell types. And so we can use those peptides that get digested right before we send it to mass spec as a normalization. And then the protein ID was done during, with a pipeline that one of our lab members developed for his PhD. So I'll show you a couple of different sort of cross-sections through the data that we're getting. I don't expect anyone to be able to read all of these things, even on this giant screen, but we're looking at data from 3 different time points now during healing, 1 week, 2 weeks, and 6 weeks post-infarction. And as a first pass, and we're also looking, at what is enriched when we do passes through this tissue with a focus on the fibroblasts, so using that alpha smooth muscle actin marker, and compare that to if we just cross-link everything on parallel sections and run that through the rest of the analysis pipeline. So we're essentially comparing what's enriched around fibroblasts versus what's on the slide overall. And So if we pull out the proteins that we have identified that we know are somehow extracellular matrix associated, and we know that because we pulled them from a list called the matrisome, where people have compiled lists of proteins that are known to be associated with extracellular matrix. What we find over here on this end, the most enriched around fibroblasts compared to everything else, is reassuringly a bunch of stuff that we know fibroblasts should be producing at this point. So collagen 1 and 3 are right at the top, periostin, fibronectin, and then several molecules, one of these you already heard a little bit about, that are associated with elastic fiber formation, so amylin, fibrillin, and fibulin, all enriched near the fibroblasts, and particularly at the early time points. The enrichment drops off a little bit at the later time points, and our best guess about why that's happening right now is because by 6 weeks, there's a lot of scar tissue, and the fibroblasts have made it all. And so the relative difference between what's right next to a fibroblast and what's everywhere else actually starts to drop off at that point in the healing process. Okay, so then another example, all of the analysis is still the same, but in this case, in order to sub-select some proteins to look at in terms of what we know about their function, we pulled out some related to immune response from innate DB. And here we're looking, oh, sorry for the erroneous label, we're looking at things that are enriched at macrophages compared to the whole slide. And there, again, maybe not surprisingly, we pull out things that you would expect to see in a healing wound and resolution of hemorrhage, for example, like a bunch of different complement components, plasminogen. We see some heat shock proteins, and one of the things that pops out at all the time points is plectin, which I mostly know as an associate with several malignant cancers. All right, so in those cases, what I was doing was pulling out subsets of proteins based on some function that they perform that we're interested in. What if we do it without trying to pre-select it all? So what we've done here is to take all of the proteins we identified and group them by pathway, and in this case, we're using pathways from Reactome. And then we're plotting what's most enriched in the fibroblasts versus what's most enriched in the regions around the macrophage, and then we're doing that at different times. And one of the things that was most interesting about this is that actually there's a lot of concordance between the things that are most enriched at the fibroblast interface and most enriched at the macrophage interface. And again, the very top hits for this made a lot of sense to us. So the pathways that jump out are related to integrin signaling, related to integrin-linked MAP kinase signaling, PDGF signaling, platelet-derived growth factor, and then other ones are more generically labeled cell matrix interaction, ECM organization, and there are several pathways that specifically relate to collagen, so collagen assembly, collagen cross-linking, and collagen degradation. And then you see that at 6 weeks, things start to get a little more interesting potentially. You start to get more decorrelation between what's happening at the fibroblast surfaces and what's happening near the macrophages. Interestingly, this group up here, which is still enriched around macrophages but not around fibroblasts, is a lot of this same cell matrix interaction stuff. What starts to pop up as being enriched now around both the fibroblasts and the macrophages at this last time point is things related to new blood vessel formation. And so that reflects another stage of the healing of the infarct in this case. Okay, so this produces a ton of data. And the other thing that we're working on as part of this project is thinking about how to use computational modeling to integrate that data and to interpret what it means for cell behavior. And we're doing a lot of the modeling in collaboration with Shane Pierce-Kotler, who talked earlier this morning, and another colleague of ours, Jeff Saucerman, at University of Virginia. This is just an example from a paper that's in revision now. And so what we've done in this particular model is we've built an agent-based model, which I knew I didn't have to tell you about because Shane already told you about it this morning, and we've embedded into those agent-based models, instead of a model of an E. coli, which is what Shane talked about this morning, we've got a model of a fibroblast. And so what's happening here is fibroblasts can crawl around in their environment, and out in that environment, they're going to pick up information on different inputs that we know are important for fibrosis, including cytokines and things like TGF-beta, which can be bound in a latent form to the matrix and then get cleaved to act as one of these matrix-derived cytokines that we talked about. And so as they're crawling around in their environment, they're picking up the levels of all of those things. And then for each fibroblast, we feed those levels into a large network of ordinary differential equations that reflect what we know about fibrosis signaling inside the fibroblasts. And so each fibroblast can run its network, its signaling network, and figure out, based on its environment, what is it going to produce? And then the things that it's producing are these extracellular matrix molecules that we're interested, like the collagens, the MMPs, and also in some cases, some of these signaling factors that can have an autocrine response. And then this is just an example where Shane's student, Michaela, we built a little artificial environment where we're giving different levels of cytokines that are either increasingly fibrotic along this axis or increasingly inflammatory along this axis. And we just turned some cells loose to crawl around so that we could understand how the cells are behaving when they're crawling through this changing complex environment. And then over here, she's plotting the lines are the levels of some of the inputs that the cells are detecting. And then with a time lag, the bars are showing what we're expecting the cells to produce. And then, of course, as these things are getting produced, here in the agent-based model, they can be deposited in the matrix, and as we talked about in the opening, be picked up by the next cell that comes along, so that as you're depositing things, or we could also have them making modifications like cross-linking. So as you're modifying the matrix, then you're affecting not just what's happening to you now, but to the next cell that comes along and encounters that modified matrix. Okay, so in summary, we said at the beginning that the cardiac extracellular matrix provides critical structural and chemical cues that are guiding cell structure and function. And then I showed you in the middle that the cell-targeted STOMP technique that we've developed with the support from the Allen Frontiers Allen Frontiers Group, and the American Heart Association, has allowed us to do that cell-targeted STOMP and show that we are enriching our mass spec analysis for proteins that are associated with cell ECM communication and ECM turnover. So this was Sunil's needle in a haystack. We've made the needle that we wanted, the cell extracellular matrix interaction, much bigger relative to the haystack that we're working in. And then finally, that we're able to build these hybrid ABM network models that give us a framework for integrating data on the cell ECM, the cell, and what I'll call cell ECM cell, where the cells are communicating with one another, but not directly through the modifications that they make in the extracellular matrix. So to conclude, just like to show the members of the team. These are the 4 PIs on the overall effort, and then a number of students and postdocs who did the work. Laura Caggiano is a grad student who's doing the animal experiments. Venk is a postdoc who's doing the bioinformatics analysis. Michaela, I mentioned, has been working on the agent-based modeling, and Bo Chang is a postdoc who works on the STOMP. And then collaborators Shane and Jeff I mentioned as well. Thank you.
We're going to break now for lunch, and people can find Jeff and have a great lunchtime conversation. There'll be buffets set up in the lobby, and you can either eat in the lobby or there's room also on the second floor and in the cafe. And we'll meet back here at 1:30 for the next presentations. Thank you.